Well, hi everyone. Thanks so much for joining this afternoon. Thanks to Hamish and Kevin for the invitation to talk about my research today. My name is Naral. I'm a microbiologist. I'm based at the University of Technology, Sydney, and I love bacteria. I'm really interested in understanding how bacteria cause infections and disease and what we can do to stop them from doing this. And this is really how I ended up doing research on honey, because we know that honey has a long history of use as an antibacterial medicine. I'm also interested in understanding how bacteria, particularly those living in our gut, can influence our health. So I look at how eating certain foods, like honey, may be affecting the balance of these bacteria living in our gut. So today I'll talk a little bit about what history has taught us about the medicinal uses of honey, what our research has shown us about the best ways to use honey as a medicine, and how all of this links into the world of bacteria. So bees have been around for many, many more years than we humans have. There are about 20,000 species of bees, about 7 of which are the honeybee. So these live in colonies and produce significant amounts of honey. Of these species, one of the main ones that we source most of our commercial grade honeys, or the commercially produced honeys, is the European honeybee, the Apis mellifera. So from this picture here, we can see a natural comb and hive. So what the bees do is they'll go off to flowering plants, collect nectar that they bring back to their hive, and they put the nectar into one of these little comb cells. Then they all work together to dehydrate that nectar, and once it's dehydrated enough, they'll cap that comb or cap that cell, and then the honey matures over time in the hive. And it seems that we humans have always had a bit of a sweet tooth, and honey has been an important dietary staple for many peoples around the world. And we often went to great lengths to get it. So in this first picture here, you can see that we've got honey hunters climbing on these really tall vines to get to the hive at the very top. Many cultures have documented honey hunting or robbing, and this involves gathering honey from wild bee colonies. So we've got rock paintings from around the world that provide some of the earliest evidence of gathering honey from these wild colonies, and usually the process involves smoking of the bees of some sort. So here in this cave picture, you can see the figure here holding a flame torch at the entrance of the hive and the bees streaming out there so as to give the person easy access to collect honey from that hive. Honey hunting is still practiced by many indigenous peoples around parts of Africa, Asia, Australia, South America, and in Nepal, which is this picture here, we've got honey collectors still climbing these bamboo ladders positioned off the surfaces or the edges of cliff faces to access these hidden caves. Now, the problem with honey hunting is that it would often destroy the hive and the bee colony. So we didn't have repeat access to that honey production or honey collection. So there was this need for a way of keeping the bees, the same colony year after year, so that we could keep producing honey. And this is how beekeeping, as we know it, was born. The earliest records of domestication or keeping of bees is shown again in ancient Egyptian art form, where the hives depicted here and in this schematic here were made from hollow logs, wooden boxes, pottery vessels, or woven baskets. So the brood was kept at the back of these hives so that the beekeepers could access the honey from the front without damaging the health of the brood at the back. The ancient Egyptians would follow the flowering plants with their hives up and down the River Nile, and this allowed for the production of large amounts of honey season after season, year after year. And we know that honey was a very important food and medicine for the ancient Egyptians. And in fact, our historic records indicate that honey has been used as a medicine by every culture that's had access to it throughout all of history. It's been used to treat various ailments, from eye and throat infections to gastroenteritis, but it's been persistently popular as a topical application to treat wounds and skin infections. So here we've got an example of an ancient Egyptian script, a prescription or a medicinal script, and this is for a wound salve. And we can see that honey is a prominent feature in this wound salve, as denoted by the picture of the honey jar there and the bee right next to it. So again, this preparation was used for the treatment of a range of different wounds, including surgical wounds, like what the ancient Egyptians used for circumcision, which is what's shown in this schematic and carving here. And why it was effective was that it would stop the infection from forming in the first place for these surgical wounds, or if an infection had formed, it would be used as a treatment to clear that infection. Although it's unlikely that the ancient cultures would have talked about bacteria and infections in that kind of way, since we didn't know about the hidden world of bacteria until much, much later. And then with the discovery and mainstream use of modern antibiotics in the 1940s, the medicinal use of honey was largely left in the past. But today we have a shortage of effective treatment options for infections, and we're seeing a huge increase globally in the number of antibiotic-resistant superbugs, or antibiotic-resistant bacteria. Now this happens because the bacteria are evolving very quickly to fight off the effects of our common antibiotics, much quicker than we can discover new ones. And we're already seeing cases of bacteria that are resistant to every single antibiotic we currently have available to us. So along with climate change, it poses one of the biggest threats to our society today. So I'll just talk a little bit about antibiotics and superbugs and how honey can help with this issue. So antibiotics are drugs that work by killing bacteria or slowing down their growth, which means that they can no longer cause an infection. And the way that they work, like in this cartoon, is that they target certain things in bacteria, like the protective cell wall or parts of their metabolism. But bacteria are very clever and they evolve or change very, very quickly, much faster than we do. So they quickly learn to fight off the effects of these antibiotic drugs, basically rendering them useless. And this is what's known as antibiotic resistance. So it's not that the drugs simply stop working or that we stop responding to them, but that the bacteria have changed to fight off the effects of the drugs. And these antibiotic resistant bacteria are what we commonly hear as being called superbugs. So when bacteria become superbugs, they hold on to what gives them the ability to resist antibiotics in their DNA. It gets ingrained in their genetic material because it gives them this competitive edge for survival. And then they can swap and share this DNA with other bacteria, which means that they can give and take the ability to resist other antibiotics as well. And what we end up with is different types of bacteria that are resistant to multiple different drugs. And these bacteria keep passing on their antibiotic resistance to each new generation, creating more and more superbugs, which then really limits our treatment options for people who do end up with these superbug infections. And it's because we urgently need alternative approaches for infection control that there is scientific interest in complex natural products with antimicrobial activity, like honey. So honey, of course, will not fix all of the issues with antibiotic resistance, but it can directly help to relieve some of these burdens, at least in the wound care sector. And despite being used for thousands of years as a medicine, there are no reports of bacteria becoming resistant to the killing effects of honey, like they become resistant to antibiotics. So honey does have a real potential to be used as a first-line antibacterial treatment for the treatment of things like wounds, burns, ulcers, and skin infections. And in Australia, chronic wounds, which are wounds with very delayed healing, affect over 400,000 people, and they're one of the leading visits for GPs today. They're also increasing in prevalence because we're shifting towards an aging population, and we have higher incidences of obesity, diabetes, and other inflammatory conditions where wounds are one of the main pathologies associated with those conditions. So although my research today will focus on the antibacterial properties of honey, there are also several other properties of honey that make it a really good wound dressing. For example, it does provide this moist healing environment. It stimulates our immune system, it reduces inflammation, and it can even help with wound odor and debriding or a cleaning action as well. We already have lots of honey-based wound care products available for use, both in the clinic and over the counter for home use as well. And these are approved by the regulatory bodies like the TGA in Australia for medicinal use. So there are lots of case studies showing that honey has worked where antibiotics and other conventional therapies haven't. And we're looking at one of these now. And sorry, I should have just given a little warning. I will have a couple of wound pictures pop up. So if you feel a bit squeamish and don't like looking at them, please feel free to look away. So here we've got a 78-year-old patient with a non-healing diabetic leg ulcer. And over an 8-month period, a variety of treatments were prescribed. So we had antibiotics to clear the infection, compression therapy to help with wound healing, but the patient just didn't respond to these treatments. Honey treatment was started, and by day 22, you can see that the infection was cleared and that wound had significantly healed. And obviously this is not the only case study. Honey's been shown to work very well on these extreme chronic wounds, but also just as effectively on acute wounds and skin conditions like eczema as well. So where there's no infection involved, but there is inflammation. But honey is still overlooked as a treatment. And one reason for this is that it's still seen as very much an alternative, not a conventional treatment. But we are seeing a shift in these attitudes in the past couple of years because we're desperate for treatments that do work. And another reason that's overlooked, and usually this is by clinicians, is that although we know it does work, we don't fully understand how it works. And this is one of the major areas that we're working on. So one of the most common misconceptions about honey is that all honey is the same. But honey is not a generic or a standard product at all. It's very complex. It has over 200 components. And in the same way that different honeys can taste or smell or look very different, they can also have very different medicinal properties. There are multiple factors that contribute to its antimicrobial activity. And all honeys will have some degree of antimicrobial activity, but not all are at levels that can quickly clear infections. So the first factor that gives honey its antibacterial activity is that it has high sugar levels, which means that it has very little water available inside the honey. And just like us, microbes need water in order to survive. So this high sugar actually dehydrates or dries out the bacteria. Honey is also a very acidic solution. It has a low pH, and this is not tolerable by most microbes. So these two factors are similar across all honeys, including the ones that we have at home and use on our toast in our pantries. But they're not at levels that are high enough to clear an infection at all. So when we're looking at honeys with really significant antimicrobial activity, this is usually from a range of different factors. The most significant one, or the most common one, being this production of hydrogen peroxide, which is like a weak bleach, and it's toxic to bacteria. So these peroxide-based honeys, the honeys with this hydrogen peroxide activity, vary hugely depending on what flowers the bees have visited to make that honey, also how the honey is being stored, how it's being processed. There's a whole range of factors that affect this hydrogen peroxide activity level. We also have some very rare honeys with high levels of activity that are beyond this hydrogen peroxide activity. And this is also linked to floral factors. And the most famous example is the Manuka honey from New Zealand and the similar honeys in Australia, where the activity comes from a different chemical that's related to the floral source. And we do a lot of research on these Manuka and related honeys in our lab. So Manuka honey comes from the Manuka plant, or the scientific name being Leptospermum plant. And what gives this honey its special or its added level of antibacterial activity is a chemical called methyl glyoxal, or MgO. And this comes straight from the floral source. So in this graph here, we're looking at the active ingredient, the level of MgO in a honey sample, in a New Zealand Manuka honey sample, and the related antibacterial activity. And if you follow the graph, you can see that as that active ingredient level increases, so too does the antibacterial activity. So Manuka honey is the most commonly used medical grade honey around the world because it is very potent and has very stable activity. And that means that we can sterilize this honey by gamma sterilization, for example, and that makes it suitable for clinical use without affecting its activity profile. So this brings me to the many research questions that we're working on in our lab that are based around this honey idea. We want to know how broad spectrum the activity of honey is. Is it equally effective against these antibiotic resistant superbugs? And will we see honey resistance if we start to use honey more, just like we do with antibiotics? Can we use honey alongside or in combination with antibiotics that we currently use to increase their effect? What's the mechanism of action of honey? So exactly how does it work to kill bacteria, and why don't we get resistance to it? Where can we find more sources of medical grade honey? If we start using it as a first-line treatment, are we going to run out? And of course, what about eating honey? So I've talked a lot about its topical applications, but are there any therapeutic or health benefits of eating honey as well? So some of the things that we found in our research over the years is that honey is very good at killing bacteria that cause infections, including superbugs that are resistant to antibiotics. And it doesn't seem to matter what antibiotics they're resistant to or how many they're resistant to. Honey doesn't discriminate. It can stop the growth of the ones even resistant to multiple drugs. We've shown that it's very effective against common wound pathogens, so bacteria that infect wounds, including things like MRSA or golden staph, Streptococcus, which is known as the flesh-eating bacteria, and Pseudomonas, which can cause a lot of issues in burns patients. Probably one of the most exciting things about honey scientifically is that bacteria don't seem to develop resistance to its killing effects. So I've already mentioned that there have been no resistant or honey-resistant superbugs reported, despite its use for thousands of years, but we also can't make them resistant in the lab either. So here are some results from some experiments that we do in the lab, and I'll talk through these. But what we do is we treat bacteria with increasing doses of honey or antibiotics and train them to become resistant to the treatment. So we train them to get used to living with this pressure, this killing pressure in their environment. And in the results here, when the graph deviates above that red dotted line, it means that the bacteria are resistant. So the treatment is no longer effective. So if we follow the black line, which is the antibiotic testing, you can see that just within 2 weeks, that graph deviates above the red line. So we're starting to see antibiotic resistance form very, very quickly. And the longer the experiment goes, so here we've got it up to 30 days, the longer the experiment goes, the more resistant these bacteria become. If we follow the blue line, which is our honey experiment, we can see that that level never reaches that resistance threshold. So even if we pushed it out to 30, 50, 100 days, we don't see honey resistance forming in the lab at all, even though antibiotic resistance forms very quickly. So we've tried this with lots of different types of bacteria, lots of different types of honey and antibiotics, and the results are always consistent. So what complicates infections even more is that bacteria usually exist in what we call biofilms. So this is when free-floating bacteria, like what we have in this graphic at the top here, come together and they form a network, and then they attach to a surface, which is what we see at the bottom of the graphic there. So this surface can be a living surface like a wound bed, or it can be a non-living surface like a catheter or another implanted medical device. So this network of bacteria then all start working together to form this gooey, slimy shield around them. And this shield actually stops the bacteria from being cleared by our body's defense system, and it also protects them from the attack of antibiotics. So of course, we wanted to see if honey could be used to treat biofilms as well. And we found that honey not only prevents biofilms from forming in the first place, but it's also effective at clearing these biofilms once it's already formed. So here I've got some results from experiments again, but what we're looking at are some images from under the microscope showing biofilms. So that fluorescent green indicates living biofilm cells, and the red indicates the dead biofilm cells. So in the first image at the top, you can see when there's no honey in that setup, we've got this really bright lawn of fluorescent green living biofilm. And as soon as we introduce honey into that system, we can see a huge reduction in that fluorescent green, which means that the biofilm is either not forming or it's being cleared very quickly by the honey treatment. And this is relevant in the clinic too, because we're often dealing with biofilm infections. So here is another case study. And this is one of the most impressive ones I've ever seen, so I always talk about this one. But what we're looking at here are some non-healing leg ulcers in an 80-year-old woman. And this patient had been suffering from these infected ulcers for more than 20 years on both her legs. The pain was so bad that she couldn't walk. Her doctors recommended amputation because they couldn't get the infection under control, despite trying every antibiotic, every kind of compression therapy, and every kind of dressing that they could think of. And they just didn't want the infection to spread anymore. So one of her nurses suggested using honey dressings as a last attempt. And you can see that within just 10 weeks, the wounds were completely healed and she had regained use of both her legs. So that infection was completely under control and the wounds had healed. She could walk again. So in this woman's case, the honey literally did save her limbs from being amputated. So we've also done a little bit of work to look at combination treatments using honey with antibiotics. And the reason we want to do this is to see if there's any synergy. So can the two of these things, a honey and an antibiotic, work together for a better outcome? And this better outcome could be something like a shorter treatment time or reduced dose of antibiotic. So what we're looking at here in these plates are agar, which is a source of food for our bacteria, what we grow our bacteria on. And the cloudy zone there, or the cloudy growth there, is actually bacteria, golden staph. In the middle, we've got this little paper disc soaked with an antibiotic. And just around that disc, you can see this, what we call a zone of clearing. So it's a clear zone around that disc, and that shows that the antibiotic's working to clear the bacterial growth around that dose. And we've got, indicated by these red arrows here, these single colonies, these little dots of bacterial growth. And these are bacteria that are starting to resist the effect of that antibiotic. So in the second square, we've got the same kind of setup, but this time we've introduced honey into that agar plate as well. And what you can immediately see is that zone of clearing around the disc is much, much larger. So the honey and antibiotic are working together to clear even more of that bacterial growth. And we don't see those resistant single colonies popping up either. So this is just an example in one type of bacteria, but we see the same kind of thing in bacteria that are growing in that biofilm form as well. When we have honey and an antibiotic together, they usually work better to reduce the dose, but have a greater clearing effect. So our PhD student, Daniel, who has just recently finished, was working on understanding what Manuka honey is doing to bacteria at the genetics or the DNA level. And this is so we can figure out exactly how it works to kill bacteria. So normally antibiotics will target one main thing in bacteria, and these are just examples of systems in bacteria that antibiotics can target. But what Daniel found was that honey is so effective because it affects many things or many processes in bacteria. So it's not just targeting one thing, it's actually targeting multiple things. Things like the way that bacteria organize their DNA, their metabolism, their ability to move around, and their ability to communicate with each other, which are all really important for establishing and continuing infections. So with this information, we'd like to use it to help design new drugs that work like honey, so they can kill bacteria without resistance. And the other angle of the project is that we want to support and increase use of honey in the clinic as a first line, not a last resort treatment. And currently, New Zealand is the main producer of these medical grade Manuka honeys, and already they can't keep up with the global demand. So this is where our Oz Honey project comes in, where we were looking at the Australian leptospermum honeys, or our Manuka equivalents. The main focus of the project was to survey Australian leptospermum honey, nectar and plants, and to see how the activity of our varieties compares to this one variety that's found in New Zealand. And we do have lots of different leptospermum species here. We've got over 80 that are native to Australia, and they grow all around the country, as you can see on that map. But we really had no idea how the activity compared to that one famous species that's found in New Zealand. So you may remember that I said the activity of that New Zealand Manuka is linked to a key active ingredient, that MgO, or methyl glyoxal. And when we started our Australian survey, we had no idea if that same relationship was true for the Australian varieties. So the first thing we did was dig up a library of Australian leptospermum honeys, and we tested their antibacterial activity and also measured the level of MgO in these honeys. And we found that same relationship. So as that active ingredient increased, so too did the antibacterial activity of that honey. And because these honeys were from an older project and had been stored in the fridge for a long time, for over 7 years, we had a chance to compare how the activity changed over that time. And we found that there was no change. So they were just as active as they were when they were first harvested 7 years ago. And this has really positive outcomes for extending the shelf life of medicinal honey products. So for the new survey, we sent a call out to all of the beekeeping associations and groups asking for Australian leptospermum honey donations. We collected over 1,000 samples from different regions of Australia. So on the map, you can see all the regions that we collected these leptospermum honeys from, and each of those different colours represents a different species. So of our 83 species that grow in Australia, we covered probably about 30 species when we were looking at their activity. Most of our honeys came from New South Wales and Queensland, which are our biggest beekeeping states. And we found that there are several key species, which I've listed here, across lots of different states that can be targeted to produce really highly active honeys, levels of activity we haven't seen before. So we mapped the locations that these honeys were sourced to look for certain regions that might be hotspots for producing these active honeys, the honeys with the high levels of MGO. So the colors on the scale show increasing levels of MGO, and what we're interested in for therapeutic use is anything that's above that yellow, orange, red scheme. And you can see on the map that we do have certain regions that are active hotspots to produce these antibacterial honeys. So because of the different levels of antibacterial activity and the medicinal benefits associated with them, there are a number of rating systems that are used on medicinal honeys. And this can be really confusing for consumers. But the very simplified explanation is that all of these numbers on honey, or a medicinal honey, refer to its antibacterial activity. So it's not about how good it is to eat or how healthy it is. It's simply about how well it can kill bacteria, where the higher the number, the more effective it is. So I'll try to explain some of the main rating systems used on medicinal honey. So first we've got the total activity, which we also call bioactivity. And this is used on lots of different types of honey from different floral sources. And it refers to the antibacterial activity that comes from all of the components in honey. So we're looking at the sugars, the pH, the hydrogen peroxide, and any non-peroxide activity as well. And a really famous example of a honey that has really high total activity is Jarrah honey from WA, which has been in the news recently as being this super honey with really high levels of activity. So what this number means, where you see this 10 plus or 15%, it's relating it back to a sterilizer that we compare it to. So we could use phenol as our standard, and that refers to like a 10% activity means that it's equivalent to a 10% solution of phenol. So think of this sort of like the SPF rating on sunscreen, where the higher the number, the more active it is at killing bacteria. Then we have the non-peroxide activity, NPA, which is also referred to as the UMF. And this refers to activity that's specific for Manuka or Leptospermum honeys. Again, this is tested using a biological assay. So we test it directly against a bacteria or different types of bacteria. But this time we remove all of the other components of honey and just test for that added or that unique Manuka activity. So NPA and UMF are actually measuring the exact same thing, but UMF can only be used under a trademark registered in New Zealand. And then finally, we've got this methyl glyoxal or MGO-MG rating. Again, this one's specific to the Leptospermum or those Manuka-like honeys. And this one's a completely different test. So there's no bacteria involved, it's a chemical test. It directly measures the amount of MGO in a honey sample. So it's looking for the level of that one active ingredient only. And this is where we get a lot of confusion, because even though the MGO and the activity level, that NPA or UMF, are connected, they're measured on totally different scales. So here at the bottom in blue, I've included a little key to show how these different rating systems or the numbers compare. So you might pick up a Manuka honey sample and think MGO 83 sounds like it's a really active honey, compared to one that says NPA 20, for example. But when you actually look at that MGO NPA conversion, the MGO 83 honey actually has no activity, and that UMF or NPA 20 honey has very, very high levels of activity. So the real question is, which honey should you buy? And the easiest way to answer that is it depends on what you're using the honey for. So if it's for a general daily use as a food or a tonic, there's no need to buy the more active and therefore more expensive types of honey. And it also doesn't have to be Manuka honey either, and we'll cover that in the second part of the talk. But the right kind of honey is very effective as a wound dressing. So if Manuka honey is being used to treat wounds and skin infections, it should be active, it should be sterile, and it should be appropriately packaged and registered as a medicinal product. So the best way to ensure this is to check that the product has a CE mark, or it's registered with the Australian Therapeutic Goods Administration, which means that it will have an OST-R number on the packaging. And usually these aren't the honeys that you buy in the supermarket or at health food shops. These are the ones that are available at your pharmacy. So we know that most people are buying honey as a food, not as a topical antimicrobial. So for the last part of the talk, I'll focus on whether eating honey has benefits for our health. So like most other medicinal uses of honey, there is a long history of use of honey as a treatment or a tonic for various gastrointestinal conditions. So here we're looking at some ancient Arab scripts showing the making of medicine from honey. And according to these scripts, this medicine was prescribed to cure weakness, loss of appetite, digestive disorders, also used to cleanse intestinal ulcers and reduce swelling of the intestines. So again, our research question was, could some of these ancient remedies be explained by modern science? And this was the focus of my PhD work, looking at whether some of these reported digestive benefits of honey were related to the bacteria and other microbes living in our digestive tract, which we know collectively as our gut microbiome. So although I did spend the first part of the session talking about really nasty infection-causing bacteria, these actually only make up a very small amount of all bacteria. So for more than 100 years, we believed that avoiding bacteria or removing them from our bodies and our environment was the simplest or the right way to stay healthy. But most bacteria actually do us a lot more good than harm. And it's just as well because bacteria are found everywhere. They're in the soil, they're in the water, they're in the air, and they exist in huge numbers, one of the most dominant life forms on Earth. Bacteria exist in communities that we call microbiomes, and these microbiomes are made up both of beneficial and potentially harmful bacteria, mostly living in harmony. And we now understand that these bacteria are essential for our survival, and they perform a range of very important functions, both in the environment and in our bodies as well. Each of us carries huge populations of bacteria and other microbes that live on our skin, in our mouths, and in our gut. So the largest population of bacteria lives in our gut. We call this our gut microbiome. And each of us has a unique gut microbiome, just like our fingerprints. So there's a lot of research now in trying to understand what it is these gut bacteria in our bodies do and just how important they are. We know that these bacteria influence the normal functioning of our bodies and our nutrition and health. They can help to build our immune systems and fight off infections and disease. They help us to digest our food, they make essential vitamins, they remove toxins from our bodies. They also influence our hormones, our immune system, and even the way our brains work. And because of these many important roles, we're really focused on creating and maintaining a healthy gut. But we still don't know exactly what a healthy human gut should look like. What we do know is that it's important to have lots of different types of bacteria, and they should be in a balance of the beneficial types and even some that are normally thought of as potentially harmful. And the reason that we want some of these potentially harmful ones is that they help to train our body and our immune system to recognize when there's an overload of the harmful ones and do something to mitigate those effects. So when this balance is off and we see a lot more harmful bacteria than good ones, this can lead to different gut diseases like colon cancer, but also disorders outside of the gut as well. Because our gut bacteria seem to affect whether we get fat, suffer from high blood pressure or allergies, and even mental health issues. And there are a few ways that this balance can change. So our lifestyle, whether we take antibiotics, and our diet. And this is where certain types of foods, called prebiotics, can help to boost a good gut balance. And this is where my research is focused. So a prebiotic is a complex carbohydrate or a complex sugar, like those found in many root vegetables, that we don't use ourselves. And that means that it reaches our lower gut or our colon, where it can be used as a food source by the trillions of bacteria living in our gut. So prebiotics are different to probiotics. Most people have heard of probiotics. Probiotics are living good bacteria that we eat, and that can be in the form of supplements, those fermented milks, and yogurts. So these introduce one large hit of good bacteria to the gut at a time, whereas prebiotics are foods that feed the bacteria that we already have living inside of us. So the main question that I wanted to answer during my PhD was if honey is a good prebiotic. We know that honey is made up mostly of simple sugars, fructose and glucose, which our bodies would absorb and use straight away. But there are some complex sugars called oligosaccharides that we don't digest, and that's what could be driving some of this prebiotic activity and explaining some of those ancient uses of honey as a digestive aid. So in my PhD, I tested this in a lab model, replicating the digestive process in our bodies to see how the populations or the balance of bacteria in our gut changed when honey was added to this gut system. So we looked at over 20 honey samples from different floral sources, and we monitored how the populations of those beneficial and potentially harmful bacteria changed, and also looked at the levels of certain compounds the bacteria were producing in this model. We wanted to understand if the activity was related to a certain honey type or floral source as well. So to sum up 4 years of research in one little cartoon, here we've got our gut bacteria. We've got both beneficial and harmful types. When we introduced honey into the system, we saw a big increase in the populations of the beneficial bacteria. And when we have this favorable type of balance, our gut bacteria break down the complex sugars in the honey to give us beneficial compounds called short-chain fatty acids. And one in particular that we were looking for is called butyrate, because we know that it plays a protective role against the development of disease that starts in the gut, including things like colon cancer. We found that honey really promoted the production of this beneficial butyrate. And although there were some differences in the ways that different honeys were working, so some honeys boost more of the good gut bacteria, some help to produce more butyrate, the overall trend was that all of the honeys we tested had some level of benefit. So we then conducted a small human clinical study. We recruited 40 healthy volunteers and asked them to eat 20 grams of honey a day, which is roughly a tablespoon, for a month. We looked at the changes in their gut populations from stool samples, and we also tested, we also looked at the levels of those short-chain fatty acids, including butyrate. So we tested 4 different honeys that we chose from that lab model, and we chose those 4 because they all had some kind of beneficial effect, but they were all doing something slightly different in the lab model that we looked at. So here we're looking at how the gut populations of the participants changed over time after that month of eating honey. So the grey lines that you can see are the responses from the individual participants. The solid black line over the top shows the overall trend for all of the participants. And what we found was that the numbers of beneficial bacteria, so we're looking at 2 types of beneficial bacteria. We've got bifidobacteria in the blue and lactobacilli in the green. And we found that the overall trend was that the numbers of both these beneficial populations went up after that month of honey consumption. We also looked at a potentially harmful group, Clostridia, and we saw that overall the trend was that Clostridia numbers went down after a month of honey consumption. Next, we wanted to check if these responses were due to a particular honey type. So here we've got the 4 different honeys, and we're looking at the effect on lactobacilli, which is one of those beneficial populations. Again, the grey are the individual responses of the participants, the solid black is the overall trend. And you can see that no matter which of the 4 honeys were used, the trend was that the lactobacilli went up. Bifidobacteria, which is that other beneficial bacterial group, again, it didn't matter which of the 4 honeys we tested, the trend was that the numbers went up. And then Clostridia, which is the potentially harmful group, again, it didn't matter which of those 4 honeys we used, the overall trend was that the Clostridia numbers went down. So the overall conclusion from this study was that all honeys we tested did have prebiotic activity, and the activity was not linked to a particular floral type. So it wasn't a certain type of honey that was driving this activity, it was just the complex sugars in honey in general that was driving the activity. So now we're running a new project at UTS, working with collaborators at the Microbiome Research Centre and at St George Hospital in Sydney. And we're looking into this prebiotic activity a little bit further. We're running a clinical study to figure out exactly how honey affects the entire gut microbiome. So we're looking at how honey affects the microbe population in the gut, also what metabolites or compounds are being produced by these microbes, including those short-chain fatty acids, and how all of these changes might be affecting our immune response, which we know starts in the gut. So our goal here is to show how eating honey can change the gut, and then use that information to target certain gut conditions or diseases where honey may help to re-establish or repopulate a healthy gut microbiome. And one example being, we could use honey to target C. difficile infection, which is a major health threat with no effective treatment options currently. So to wrap up everything that I've talked about today, even though there are some very nasty bacteria out there that cause bad infections and disease, they only make up a small part, and most bacteria are actually beneficial to us. So we can learn to work with bacteria to improve our overall health. And this is especially true for the ones that live in our gut, because they have such a profound influence on our nutrition and health. And we can nurture these by feeding them the right kinds of prebiotic foods. And of course, honey, which has been used as a medicine since ancient times, does have many scientifically proven therapeutic benefits. It can be an excellent prebiotic when taken orally to promote gut health, and also very effective topically as an antibacterial treatment for wounds and skin infections. So by understanding all of the different bioactive properties of different types of honey, we can really tailor the way we use honey as a medicine depending on what disease or what infection we want to target. So I'll just finish off by thanking the many, many researchers who have contributed to all this work. The biggest thanks, of course, always goes to the Australian and particularly the New South Wales beekeeping industry, because they are wholly supportive of the research we do and always sending us honey samples to test, to answer our many questions. The funding for our project comes from the New South Wales Government, AgriFutures, under their honeybee and pollination program. And we also work very closely with our industry partners, Comvita and Capilano Honey as well. And of course, thank you all for your attention today. And I can see that we do have some questions in the Q&A.
Thank you so much, Niraj. Yeah, please just fire away. We can work your way through the Q&A, and if anyone has any questions, just type them in there.
Okay, so I can see Peter has asked, is there any research on honey treating shingles? Yes, there is, or COVID-19 is the other one. There is a lot of research on honey treating lots of different things. In terms of the COVID, there's not been research yet on honey and COVID, but COVID is a virus. So a lot of the work has been focused on mainly bacteria. There's been extensive work on fungi as well, these two types of pathogens. There's some work on honey as an antiviral, but the research indicates that it's not very effective, or there needs to be a lot more done. They're not very controlled. And often there's not much of a difference between a placebo treatment and honey for viruses. So a lot of the work has really been concentrated on bacterial pathogens where it is very effective. So any kind of topical infection, it is very effective against if it's bacterial, and even fungi, but it depends on the type of honey we use. So Carl has asked, if you eat a lot of honey, would it harm your gut biota? Also, if you use honey as a topical dressing, does it harm normal skin biota? And if so, what are the effects? Okay, this is a really good question. So it depends on what you mean by a lot of honey. We've got to remember that honey is a very rich source of sugar. So anything that you eat in excess, especially sugar that you eat in excess, will have some kind of negative effect. In terms of the gut microbiota, because we're looking at the complex sugars in honey that reach the gut, they're the ones that reach the lower gut, and they're the ones that have an effect. They have a positive effect in the gut, but if you're eating heaps and heaps of honey, like, you know, we tested a tablespoon of honey a day, and I think even that it's a realistic dose, but might be a little bit too much depending on how much added sugar you have in your diet. And we saw a beneficial effect, but I think anything more than that, we're probably looking at excess sugar consumption. And in terms of your skin biota, our skin microbiota are very tolerant to low pH and high sugar. So it doesn't have an adverse effect if we use honey as a treatment on our skin. Susanna has asked, why chemically, biologically is honey so effective in healing? Why do the superbugs not develop resistance? Yeah, a good question as well. It is very effective because the honey actually targets lots of different things in bacteria. So it kills them in lots of different ways. It doesn't just target one main thing. The way that resistance works is bacteria know what their weakness is. So if an antibiotic is targeting a certain thing in bacteria, they know that target spot and they work to either flush out that antibiotic or to block that antibiotic from entering that target. But because honey targets so many different things at once, the bacteria just get overwhelmed. They can't fight off all the effects, and that's why we think it's so effective. BK has asked, is there any prebiotic activity in normal honeys? So the honeys that we tested in our prebiotic study were just honeys that we sourced from beekeepers. So they're the main honeys that we produce in Australia. They're not Manuka honey. I did test our Manuka honey sample, and actually the Manuka honey had the least prebiotic activity of all of them. So the honeys that we did look at were the eucalypt honeys, and we also included some supermarket honeys as well. And it didn't seem to matter which of the honeys we used, at least in that lab model, we did see that all of them had some kind of benefit. And we think it's because there is a lot of different types of complex sugars. Even though they're in small quantities, there's lots of different complex sugars, and they're all doing something positive for our gut microbes. So we've got Andrew from the UK. What are your thoughts on synthetic honey alternatives? For example, Sergi honey that's being developed at the University of Birmingham. Okay, so the synthetic honeys is something that we also have been working towards. The issue with using a natural product is that it is a natural product. It's highly variable. So even if we keep targeting the same honey from the same beekeeper, same region, we will have seasonal variation, we'll have variation with the climate, rainfall, and it makes it really hard to create a standardized product. So since we started doing research, there's always been talks with pharmaceutical companies about developing an artificial honey. So identifying those bioactive ingredients and then figuring out what of those bioactive ingredients is doing which function, and then putting them together and creating this artificial honey. The issue is that something about that honey complex is what helps honey work the way it does. So when we break apart all these bioactive components, it's not as effective as the whole honey itself. So we actually think that the different components in natural honey or whole honey or raw honey, whatever terminology you want to use, sort of works together in synergy to create the bigger effect. So we're still working on that synthetic honey angle, but I think using whole honey and just being aware that there are different bioactive properties and different uses is probably a better way forward in incorporating honey in modern healthcare. Gemma has asked, what type of research has been done re honey improving mental health? The answer to that very simply is, from what I've seen, not much at all. The mental health angle comes in when we're looking at the gut health. So we know that so many things are linked to our gut health, and anything that we infer about mental health is related to the gut health. So this area is still very, very new. I was one of the first people who looked at eating honey on gut health. So there's been not really much done in this field at all, but stay tuned. I think people are getting on this bandwagon. Christopher has asked, if you put an 80% sugar water solution on something, wouldn't the bacteria use it as a food? Why don't they eat honey? Why don't they eat honey as a food, or is that fungus? Okay. If you put an 80% sugar water solution on something, yes, the bacteria can use it as their food source, as their carbon source, but usually 80% is very high, and it causes the bacterial cells to lyse or burst open. So high sugar and high salt actually can cause bacteria to burst open. With the honey, because there's other antibacterial components in there, it acts way quicker than, it's much more effective than using an 80% solution. So we would use 100% honey or almost 100% honey when we use it as a topical application, but we really only need something like 5 or 10% solution to inhibit or stop the growth of bacteria. And that's because of the other bioactive components. With fungi, it's a very good point. Fungi thrive off high sugar, and they can actually tolerate much higher concentrations of honey. So it depends on the type of honey we use for fungal infections. And the ones with very high levels of hydrogen peroxide activity are much more effective than those Manuka type honeys, for example, when we're dealing with a fungal infection. So Caroline has asked, does heated honey lose its good properties? The simple answer is yes, it does. So if you're using honey as an antibacterial and you heat the honey, or if you even put a spoonful of honey in a hot cup of tea, you'll lose most of the activity that it has in terms of the antibacterial activity, because most of that comes from the enzyme that produces hydrogen peroxide, and an enzyme is very sensitive to heat and light. So if you're a beekeeper and you're storing your honey tanks or your containers or vats under direct sunlight or in a really, really hot shed, and that activity is from hydrogen peroxide in your honey, then you will lose all of that activity. In terms of eating honey for gut health, this comes from a different component, which is the complex sugars, and these are very, very stable. So when I was doing my experiments, I accidentally autoclaved my honey, and an autoclave is like a sterilization oven where it goes up to 112 degrees and it's steam sterilized. And then I thought, well, I've autoclaved them now, I should just test to see if they still have prebiotic activity, and they do. So those complex sugars are much more stable, much more resistant to being heated. So if you're using it for antibacterial, don't heat it. If you're using it to eat, it doesn't seem to matter if it's heated, but just to retain the other vitamins and enzymes, it's always best to not heat your honey. Can MGO be synthesized for medicinal use? So this is from Carl, instead of relying on Manuka honey. Yes, absolutely, MGO. There are lots of synthetic versions of MGO, and they are used in some kind of infection control. The issue with MGO is that often we need much, much higher levels of MGO compared to what's found in honey to have the same level of clearing. So we've done experiments, Daniel, our PhD student, has done experiments comparing pure MGO or chemical MGO to Manuka honey. And we need much, much higher concentrations of MGO, like 5 to 10 times more than what's in the Manuka honey. And the other issue with MGO is that it's actually toxic to human cells. So if we were using it directly on human cells, it does have a bad effect on our healing and the ability of our cells to be able to repair or regenerate. So something about honey, again, MGO in the context of that honey has a protective effect. So we don't see the negative aspects of MGO, but together it works for not just infection control, but also the wound healing aspect as well. So Peter has asked, there's been a lot of publicity about adulterated honey. Is it really bad for us? What has been done to stop this practice? There has been a lot of news lately about adulterated honey, and that refers to when honey has been supplemented with the sugar syrups, like things like high fructose corn syrup, for example, or it's a sugar syrup and there's no actual honey at all. In terms of whether it's bad for us, it's another source of sugar, so it's not really any different to having high fructose corn syrup in any other form. But in terms of what it's doing for honey and the honey industry, it is a really negative outcome. You know, when you have honey and you think that there's some sort of benefit associated with it, but you're getting just a sugar solution, it's not giving you that same benefit. In terms of what's being done to stop this practice, in Australia, we do actually have very strict rules about what can be called honey, and we do have a good testing system to make sure that what says 100% Australian honey is 100% Australian honey. So if you're in the supermarket and or at a farmer's market or wherever you are buying your honey, if it says 100% Australian honey, you can trust that that is Australian. A lot of the publicity around adulterated honey comes from imported honeys where they don't have as strict regulations about what can be called honey, and they don't have the food standards like we do in Australia and New Zealand. We are also looking at lots of different ways to detect adulteration. So honey is like the third most popular adulterated food in the world. So at the moment, we look at the sugar levels and the ratio of different sugars to say whether it's been adulterated or not. But there's a whole bunch of investment in looking at other chemical tests and biological tests as well to ensure that what we call honey has come from a plant source and made by bees. So David asks, if honey is heated, for example, in your tea, do you think there's a difference in prebiotics? So in terms of those complex sugars that are giving the prebiotic activity, probably not. They don't seem to be affected by heat. If you're looking to have honey in your tea and you want to retain the antibacterial activity as well, I would just recommend let it cool down. So just don't put it in when it's boiling, let it cool down to drinking temperature and then add your honey at that point, and you'll get both the antibacterial, the anti-inflammatory, and that prebiotic effect as well. Matthew says, what would you recommend for people with colon cancer? A really hard one for me to answer because I'm not that kind of doctor. I'm not a medical doctor, but some of the work, there's a lot of work looking at prebiotics in general, to not just honey, but prebiotics to boost the levels of butyrate. So lacking butyrate seems to be a really big pathology with colon cancer. So there is a drive to boost the diet with things that will help to produce butyrate. Probably best to speak to a specialist about what kind of prebiotics can be incorporated, or if diet can be modified to you know, boost that healthy gut microbiome. So Chris asks, should I put honey on hot muffin or toast? Yes, absolutely. With peanut butter, even better. Who measures the honey so they can be given one of the antibacterial ratings you mentioned? Is it done by a reputable independent organization? A really good question from Chris. So we measure the honey in terms of our study. For 5 years, we did offer honey testing for beekeepers, commercial, hobbyist, amateur beekeepers. It didn't matter who they were. We did offer that testing. There's another research university at the Sunshine Coast that also does the testing. There are a few labs, commercial labs, who do the testing. And at the moment, we're working on building an association, looking at doing testing for lots of different types of honey. So if you're looking at Manuka honey specifically, there is already an association formed called the Australian Manuka Honey Association. And they have sort of like that tick of approval. They have a list of labs that they will recommend to say, you can get your honey tested here. And it gets that Australian Manuka honey approval to say, you've got it tested at a reputable lab. In New Zealand, they already have that industry up and running, but we're working on that here, just for the Manuka honey at the moment, but we're working on a bigger scale as well to look at the other active honeys. Gemma asks, can we access the slides after the lecture? How can we keep up to date? Yes, I will make the slides available. I think the lecture's being recorded anyway, but I will also email a copy of the slides to Kevin and Hamish. If anyone's interested, I guess we can circulate that around. I've got my email up in that corner. If you want to shoot me an email, I do have a website that I'll be updating, the Oz Honey Project website, and I'll have all of our latest publications and, you know, our research in general up there. But if you just shoot me an email, I can put you in touch, or we'll get you connected with the website and put you on our subscription list as well. Rachel asks, are there studies for locally applied honey to wounds? Is local application recommended for minor wounds? So in terms of wound treatments, I would always recommend using the proper medical grade honey that you buy at the pharmacy. And the reason for that is that it is sterile. So it's been sterilized. There's no nasties that you're introducing. So honey is a natural product. There will be little bits of pollen. There will be other bee bits and things that are in the hive as well. And especially for a wound that's already a compromised skin space, you don't want to be introducing any foreign bodies in there. So the sterilized, you know, highly active honeys are best for that use. But if you're out camping and you fall over and get a graze and you've got some honey in your bag and you put that straight on, it will definitely help with some cleaning action and probably stop an infection from forming. So in a pinch, yes, you could use it. But if it's to actually treat wounds, then I would always recommend the pharmacy one. Carl says, does the peroxide activity of honey release cancer-causing free radicals? The short answer is no, unlikely, because it's such a low level of peroxide and sustained over such a long period of time that it doesn't seem to have the negative effect that many of these reactive oxygen species has. But there's not really been that much work looking at the effects on human cells. So probably something, yeah, that needs to be looked into further. And then Lawrence says, apart from containing the most beneficial varieties of honey, how do the clinical and over-the-counter creams differ from pantry honey? So the main difference is the levels of activity and the sterilization. So in the clinical honeys, and the over-the-counter ointments and creams, we know exactly the honey source. We know where it was, who it was sourced from, when it was sourced, the level of activity, and that it's been sterilized. Whereas your pantry honey may be a mix of honeys. It may have other bits in there. When I say other bits, I mean things like pollen, plant bits, bee bits in there that haven't quite filtered out or been sterilized. In terms of eating honey, like there's the medical grade Manuka honeys that you can also buy and eat, or your pantry honey. I mean, it's up to you what you like the taste of and what you want to spend your money on, but I would say just go for the honey that you like eating, and it'll probably have some kind of benefit for your gut.
Wow, that's a lot of great questions from everyone. Thank you so much for the questions, and of course, thank you so much to Neural for answering them. If no one else has any other pressing questions, we'll wind that up because we've gone slightly over time. Thank you so much again to our speaker, Dr. Nurul Chokchetin.
Thank you.
We are recording. We'll make this available on our YouTube channel, hopefully in the next couple of weeks, as well as previous webinars. And yeah, if people are interested in the slides, we'll get in touch with Nurul. You can get in touch with her directly or with us.
Of course.
The other thing to mention just quickly is we're going to wrap up this current round of talks, but we will be back. We're still figuring out our schedule for later in the year, but hopefully quite soon, possibly mid to late August, we'll start another round of talks. So stay tuned and we'll be in touch again soon.
Thanks, everyone.
Thanks, Nurul.
Thank you. Bye, everyone.